Well, welcome everyone. Thanks for joining the webinar today. My name's Peter Godden. A lot of you will know me, I hope, and I know a lot of you guys. I see the list of who's here, 18 people from across the country. So good diversity there, and that does imply that this is a bigger problem than just South Australia, but it was the biggest problem during vintage we saw during vintage this year from several South Australian regions, particularly Langhorne Creek, parts of the Adelaide Hills, some parts completely unaffected, and likewise in the Riverland. Sporadic reports of little bits in the Barossa, but since we've been talking to people since, certainly the problem with scale and vineyards, which is the root cause of sooty mould, is more prevalent than a lot of people I think realise quite recently. So sooty mould red winemaking trial, we wanted to see the effect on the sensory attributes, chemistry, and the processing, importantly, of Shiraz wine. Some similar work was done here about 3 years ago with some Pinot Gris by Marcel Esseling, and similar results as we'll see as we go through this presentation today. So what is it we're talking about? This is a substance called honeydew, a sweet, sticky substance, excreted by sap-sucking insects. And you can see some quite small scale insects here. They tap into the vine, and from the vine they excrete most of the polysaccharides and harvest the nitrogenous compounds for themselves. So they excrete most of the sugars in this substance called honeydew. And pretty quickly the honeydew gets colonized by molds. And this is a photo taken by Jenny Venus of the Landmark Company. We acknowledge Jenny for her work here and use of her slides. She's done an awful lot of this work over several years in her own time. And as you see in the dates of some of these photographs, she's been working on this for a few years. So it's colonized by these molds. If you do an internet search for sooty mold, you'll find a species called Capnodium comes up pretty readily. The fruit for this trial, we found Capnodium back in January, or February rather, that was positively identified by Barbara Hall here at Sooty. But samples we took about 4 weeks before vintage, there was no Capnodium, but they couldn't positively identify what was there. But anyway, it's generally called sooty mold for obvious reasons. The honeydew falls onto all parts of the vine and is colonized by the sooty mold. And by the time we took these photos, most of the badly affected leaves had fallen. We've seen photos and had strong anecdotal reports of vines or parts of vineyards that get so badly affected, photosynthesis appears to stop and the vines defoliate. So the long-term effect of scale infection and sooty mold is much more dramatic than just the results of this trial. Medium-term vine decline, lower sugars, lower yields, lower vine growth, lower leaf areas that can happen after a couple of years. This is some Chardonnay fruit from the same vineyard we took fruit from this trial, and this fruit was made into wine as well, and I'll talk about that as a separate trial towards the end of this presentation. This is fruit when we harvested it for our trial, and you can see the sooty mold is very obvious there. All parts of the vine, and even underground, on the ground rather, where the honeydew drops onto the ground. We don't believe this was necessarily washed off the vines, so it can colonize wherever the sooty mold, wherever the honeydew drops. And we don't believe that this was washed off. When we were in the vineyard 2 and 4 weeks before, with a wet thumb, you could wash some of the sooty mold off the grapes, but by the time we harvested it, you couldn't do that any longer. And as I say, it's caused by scale. Here's some bigger scale. Some of the scale I saw in the vineyards this year were probably 7 or 8 millimeters long. I've never seen anything quite like it, and very large numbers. And mealybug is the other insect, sap-sucking insect that can produce honeydew as well. We found a few mealybug when we harvested the fruit, but the scale was clearly the bigger issue in these vineyards this year. So what did we do? Marcel Esseling and I harvested 2 batches of fruit off the same vines. These vines had upper and lower cordons, and because the honeydew drops with gravity, the upper cordon was comparatively clean and the lower cordon was quite badly affected. So we had 2 batches of fruit control with no sooty mold. We inspected every bunch pretty carefully and excluded anything with any other indications of mold or insect damage. So although there might have been a little bit there. and then a sooty mould-affected batch of grapes. And before winemaking began, each of the individual bunches of sooty mould-affected grapes was individually assessed by Marcel. Thanks, Marcel, for doing that, for degree of infection. So there's one that might have perhaps 10 or 15%. There's one that's probably 50% plus. Here's one that may be 80, 75, 80%. So the assessment was through the whole batch that we had a bit over 50%, 53% overall sooty mould infection. So the two batches of fruit were destemmed and crushed to create two batches of must. And then we blended back the sooty mould affected must into the control unaffected to create three treatments, and we had duplicates of each. So we had the control unaffected, and we blended back enough to create a 5% sooty mould treatment and a 25% sooty mould affected treatment. As I say, we had duplicate fermentations of each. Now, at destemming and crushing, there are obvious apparent colour differences between the must, with increasing red colour with increasing sooty mould infection. However, I took samples down to the lab pretty much straight away, and Karen Binden and Stella Casalo, they span them down and then did phenolics analysis that same afternoon. The supernatants after centrifugation didn't show any apparent colour difference, and none was shown in OD520 red colour or anthocyanins with control and 25% infection, and we did both replicates of each treatment. So that increasing colour, apparent colour at crushing is interesting. It does imply that the grape skins were breaking down during that process, and therefore that the sooty mould had somehow infiltrated the grape skins. But apart from that, there was no other difference we could see in the musts at crushing. This is the means of the two reps of each treatment. Apart from a slight decrease in bricks, and we think this is probably real, though it's very small. These vines weren't badly affected until this year. As I said earlier, that we've had very good anecdotal evidence that infection with scale and mealybug leads to vine decline and much lower sugars within 2 to 3 years. Importantly, we couldn't see any difference in the nitrogen measurement. So although it does look as though the mould infiltrates the grape skin to some extent, it doesn't appear to use the nitrogen. And as we say, that extracts nitrogen compounds out of the sap and excretes the sugar. We analysed the must for lacase and ochratoxin A, a known carcinogen associated with some moulds. Lacase obviously associated mainly or wholly with botrytis. We don't know anything else that really makes it, but none were detected. And no other difference were observed during fermentation. Certainly the fermentation rate from measuring them twice a day appeared to be exactly the same. No differences apart from that increased apparent colour in the sooty mould affected ferments in the first 48 hours or so. And all 6 malos finished at the same time. So this is a table. I'll show you a few from a paper we're finishing off and we'll publish in the next few weeks. So there's a lot of data, so I'll just highlight a few things in each. First of all, as you can see, the alcohol at each time point, there is a trend to decreasing alcohol with increasing sooty mould in line with that lower sugar in the must, which I showed you earlier. Importantly, at all time points, there's no difference in VA. So this, whatever moulds are involved, they don't produce extra volatile acidity, which is a question that lots of people ask. The sulfur measurements are interesting. I had to make the call here before bottling. We added the same amount of sulfur at each point to all the ferments, all the treatments, and whether or not to add more sulfur here, because there was lower free sulfur quite clearly in the 25% treatment before bottling, but I deem this a pretty healthy level of sulfur, and there it is post-bottling. Adding more sulfur would affect how these wines age, and we want to follow them. We've got enough wine to do at least one more sensory in about 9 or 10 months' time, and hopefully another one 9 or 10 months after that, to follow them over 18 months or more. And by adding more sulfur, we would have influenced how they age, and we really want to see what effect sooty mould has, I guess, longer term. But taking out some of that sulfur data, it is interesting that this is a measurement I really like. We came up with, when we did our closures work, the original closure trial, the ratio to free to total sulfur, because I think it tells you a lot about the wine and a wine's history. And there is this trend to a decreasing ratio free to total with increasing sooty mould. Although these differences are small, and we've got to say these ratios are extremely healthy compared to the published data that we published again every 5 years, 2 years ago, very, very healthy ratios. I reckon that this is probably real and will have some long-term effect on the aging of these wines, but again, it's marginal, commercially of no commercial relevance, but probably real. In terms of phenolic analysis, the 5% treatments tend to be lowest for some measures. We can't really explain why that is. It should be noted that we only really had the funding to do duplicates, not triplicates or quadruplicates of the ferments as we would ideally have done. So no statistical analysis variance is possible with just 2 reps. But interestingly, there are some differences. Non-bleachable pigment, which is the formation of tannin and anthocyanins together to form what were once or still called pigmented polymers, pigmented tannins, and consequently an increase in the hue. As that forms, you lose the red colour and get proportionally more brown colour, but just marginal at this stage, but that does appear to be real. And that may be connected to change in chemical age index 1, although we can't be confident that these differences are real, but it appears they are, but certainly there does appear to be a trend in these 2 measures. And therefore, if you look at the amount of anthocyanins, they are lower, certainly with sooty mould, because they would have been incorporated into these larger non-bleachable pigment molecules, so that all makes sense. So that means that the wines, perhaps with sooty mould, are very slightly more developed, but this is a much more stable, non-bleachable pigment is more stable colour than anthocyanins, so it could well be seen by some people as a positive, and I'll show you the colour of the wines in a moment. The colours were pretty good and no obvious colour difference to the naked eye. So we conducted formal sensory later, but first I'll talk about the 3 informal sensory sessions we conducted. 2 were on the 24th of May, and that was approximately 1 month after SO2 was added after the end of Malo, but before bottling. So we did the first one with 5 AWRI staff, including myself, all tertiary qualified winemakers, and 2 of the winemakers up at the University of Adelaide winery next door, which a lot of you would know. And then there's the 3rd informal sensory was with the industry working group on the 25th of July, and there's a couple of people listening in today who are here for that tasting that day with 8 industry personnel. So they're the wines when we did that first sensory up in the sensory lab next door. As you can see, the colours are all pretty consistent and pretty good, although, as I say, with that 5% treatment, the one in the middle, they appear to be lower for some of the measures, but no obvious differences here observed by the tasters. So very importantly, in all of those tastings, no taint was detected. No differences in colour were detected with the naked eye. 5 of the tasters perceived a very slight increase in astringency as the degree of sooty mould fruit in the ferments increased, and I was one of those. We didn't see that in the formal sensory, but then we weren't necessarily looking for any particular differences, just if there was a difference overall. But again, any perceived differences were described as marginal and would have been no commercial relevance. And again, the informal sensory done with the industry working group on the 25th of July, no taint was detected. Most tasters agreed that 5% treatment was lighter in colour this time than the other 2 wines. And again, any perceived differences were described as marginal. But more importantly, we did formal sensory with 35 experienced AWRI tasters on the 1st of August, and counterintuitively for sure, we can see no difference sensorially between the 25% treatment wine and the control. There was, however, a significant difference with the 5% treatment. So we had to find out what that difference was. So we went on to do a full descriptive analysis with our trained external sensory panel. And these are extracts from the sensory report I got from the guys in Lee Francis's team. And the differences were for rubber sulfide and fruit aftertaste. The 5% infection treatment scored highest for rubber sulfide and lower in fruit aftertaste. So that was the significant difference that people saw in the formal sensory, the triangle tests. And I did do a copper fining trial and find all 3 treatments at a very low level. That was assessed by 2 or 3 people before we did that fining, but obviously not quite enough for the 5% treatment. So from the report, reductive winemaking artifact appears to be the main driving factor between the wines, irrespective of treatment. Certainly nothing to do with the sooty mould itself. Now, this is a slide I showed you earlier. This fruit was made into wine in 1 tonne lots with unaffected fruit from the same vineyard as part of the Wine Australia funded regional program, Adelaide Hills and Langhorne Creek. Wines made down at Bremerton Wines. And we've done formal sensory on those wines as well. So there was Chardonnay from this vineyard and Shiraz from a different vineyard just up the road. And again, doing formal sensory, we cannot see any difference. And again, as a winemaker, I'm sure a lot of you say, well, surely that fruit would have to make a wine that's different or tainted to clean fruit. Well, we can't see the difference. We've got plenty more of this wine, and we do be doing sensory and chemical analysis on it periodically over the next 2 or 3 years. Importantly, does sooty mould cause any problems during wine processing? We looked at filterability, wine yield, degree of settling, and the volume of lees. Obviously, those third and fourth things are connected. So after I racked them before bottling, I brought the lees, as it was then, down to our lab and put them in these big measuring cylinders. There's a little bit of re-fermentation in this one of the 5% treatments, even though enzymatically it was sugar and malic dry. So we left them for 4 weeks to settle. And I hope you can see here the difference, the interface between the lees and the clear wine on top. So I racked off this clear wine. So on the next few slides, I talk about the amount of wine, the wine yield after the second racking. So this was the second racking, and this was the lees that we did more work on. And all the way through, we measured in detail how much wine we lost, or the wine yield. So this was from our little cross-flow filter for each of the treatments to go into the calculations. So I should have said at the start, these were 28 kilo ferments to start with. So again, going back to does sooty mould cause any wine processing problems, there was no difference in practice, and they were easy to filter compared to a lot of the other research wines made this year through our new little cross-flow filter. We had done filterability tests in the lab here, and no clear results, I've got to say, and that's possibly to do with the volume that we're trying to use, a 750ml bottle. And reading the couple of references that exist with the filterability test, you have to question, I think, the usefulness of it in this context, because in practice we saw that they were very easy to filter compared to a lot of other wines made according to the same protocol. And that protocol is a very strict red winemaking protocol. The wines were plunged twice every day in the same pattern, using the same plunger. Wine yield after the 2 rackings, there's no clear trend. Degree of settling and volume of lees obviously connected. There appears to be a negative effect on settling with sooty mould. And the sooty mould, there was more wet lees, that's lees after that second racking, lees solids when we span those down in a centrifuge, and dry weight of lees when we then desiccated them in an oven. But importantly, very little difference between 5 and 25% sooty mould treatment. So it appears that once you've got a bit of sooty mould there, any effect on settling or lees is going to be there regardless, which is quite an interesting observation, if indeed it's real. But as I say, these were just duplicate 28 kilo ferments. Another table from the paper that we published, so I'll just highlight a few things again. So the volume of wine recovered after 2 rackings, the sum of each of the 2 28 kilo ferments. The wine yield, as I say, no clear trend. The volume of wet lees, some increase certainly with sooty mould, an increase in the percentage. So we're seeing about 10% extra lees. For a small producer, that might not be of any consequence at all, but for a big producer, and some big producers process a lot of this fruit this year, that's a lot of extra lees to have to deal with. Lees solids after centrifugation. So there's, again, a bit of an increase, perhaps 10% increase again after centrifugation. The volume of wine removed increased fairly substantially, as you can see. Spinning them at our big centrifuge, it's not just the RPM, which is about 8,000, but it's also the G-force created. Not surprisingly, you can get a lot of wine out. So we really did dry them out. There is a bit of an increase, very slight, in the amount of wine we did get out, but not surprisingly, what we were left with was pretty dry anyway. And we then desiccated them in the oven. And you can see quite a significant increase in the dry weight of those solids after desiccation. And again, a bit of an increase in expressed, got to be careful expressing, I guess, weights versus volume, but that's what we've done here. Some increase in the dry weight of the solids. So here's some photos. I still kept these after desiccation, control 5% and 25%, as you can see, and looking down on them, I hope you can see, when I was working with them, there was a clear difference. The control leaves are very dry and crumbly and very sort of particulate. These were quite pasty. You can see their marks from thumb mark or whatever, pushing them into these containers or with the spatula. And that's remained even after desiccation. So it does appear there is a difference in the nature of these lees. So what is that difference? So I thank Karen Binden here, a lot of you will know, works in our phenolics team, and Stella Casala, who did a lot of the analysis. I'm trying to quantify using an existing method we've got, the difference in lees, and they can say something, a difference, composition of the lees derived from yeast and from grape. So total polysaccharides has increased with increasing sooty mould. And if we look at grape derived, that's also increased, as has yeast derived. Now that's interesting, more grape derived polysaccharide and yeast derived, it means we must have had more biomass of yeast, or this is the biomass of the sooty mould itself, because talking to one of our biosciences team members just earlier on, Christian Varela, the basic building blocks for cell walls with yeast and moulds will be the same. So you're probably getting the yeast-derived mannose and glucose from the sooty mould itself here as well. Why would you have more yeast? Well, it's possible that the difference, because the difference we can see here doesn't quantify the whole extra amount of lees that we see, could be extra proteins. I'll come back to that in a moment. So breaking down where those extra, exactly what those extra polysaccharides are, there's the increase of the total polysaccharides, as you can see, not linear, but very clear increase with increasing sooty mould. Mannose and glucose are the yeast or sooty mould derived polysaccharides, and an increase there, and an increase in the plant derived from the grapes. But as I said, this is not enough to account for the entire additional lee solids that we saw. There's surprisingly little in the literature about lees and the composition of lees, and this feeds into something we're looking at doing in a trial with a company in the coming vintage, quantifying the amount of lees and what is recoverable. So we suspect from based on papers that have been published that other materials just ill-defined plant fibre plus lipids and proteins. You would expect the pathogenesis, PR proteins, to increase with mould infection, a pathogen. That's what the plant does to protect itself. The PR proteins are responsible for protein haze in wine, so they're not generally degraded by yeast, but it's possible there are some that are degraded by yeast, and that might account for a higher biomass of yeast in the ferments. But again, it's, we're not sure about that, other than to say the yeast, the lees are different, and there is a higher degree of yeast or fungal derived polysaccharides in those lees. This is a recently published paper looking at the amount of lees from thermovinified red musts. And in this paper, they found 8 to 5% extra of this sort of material. In our study, it's 11 to 12.5%. So it does appear that we have extra lees which are not clearly defined, but important if you're processing large quantities of this fruit. Now, just a word of caution when interpreting these results. Yeah, we went to great lengths to have clean, and I've got a question, desiccated sooty mould, because you couldn't wash it off the grapes when we picked it, you could earlier on, and without any other fungal infections present. This might be an atypical situation, and consequently, you should exercise some care when interpreting these results. For this trial, it's good that we only had sooty mould, because that's what we wanted, clean, if you like, sooty mould. Desiccated, well, it appeared to have dried out. We had 4 fantastic weeks of weather before we picked this fruit without any rain and good, clear, sunny days. it's quite possible under other conditions that other moulds would have proliferated and you might have got a different outcome. But regardless of what the fungi are that cause this sooty mould for our trial, we can say pretty categorically, as you can see, there are very, very few differences in the chemistry or the sensory properties of these wines. We don't have time today to talk about managing scale in the vineyard. As I say, it is a major issue, and more we're hearing in different regions as we move into this current vintage or this current season, that there are pockets of it in many places, and those pockets from anecdotal evidence tend to grow pretty rapidly. So we'd point you towards this webinar that Jenny Venus did for us a couple of years ago, which is still pretty current, and which she talks about some trials with some other with various vineyard treatments to try and control the scale and mealybug. Jenny is very generous with her time, although it's a very busy time of year for her. I'm sure she'll be willing to talk to you one-to-one if you've got any further questions. Acknowledgements to thank my colleagues Marcel Essling, who harvested the fruit for me, has worked on this throughout the season and did the work with assessing the sooty mould infection in those bunches of grapes. Karen Binden and Stella Cassara of our phenolics team for their advice and doing the analysis. So say Jenny Venus at Landmark, who's working with Richard Hamilton in the regional program up in the hills and Langhorne Creek, and has been working on this problem for several years. And Phil Riley and Tim Smith of CMV Vineyards in Langhorne Creek are happy to be named as the source of the fruit for this trial. Very generous with the use of their vineyard for the trials, including some of the ones that Jenny talks about in that webinar I just pointed you towards. And we thank and acknowledge Wine Australia for their funding. This trial was funded through funding for our help desk project. And when I say we only had funding for 2 replicates of each treatment, that's the call that we had to make with the funding that we have available each year. Would have been great to do more ferments, but that just wasn't possible. So this work is supported by you, Australia's grape growers and winemakers, through your investment body, Wine Australia. And the AWRI is a member here on the Wake campus of the Wine Innovation Cluster with the University and CSIRO. So thank you for your time. We now have time for questions. And we do have one question, and that is, would I have expected a different result if the wine had been on skins for longer? I think that's a good question, and my answer is I wouldn't have expected to see a taint. I think the fruit was crushed on the 27th of March and pressed on the 6th of April. So it was on skins for about 10 days. I think if there was a taint, we would have seen it in that time. I think that what would have happened if you'd left it longer, and I've got some experience with long maceration winemaking compared to standard maceration with the same fruit. What happens over time is that, especially with the breakdown of the skins we believe we saw with the sooty mould, that those skin fragments will actually absorb colour, and in the end that will drop out in these extra lees, and you probably will end up with less colour over time if you left this on skins for longer. That's what I believe would probably happen. So as I say, crushed on the 27th of March, pressed on the 6th of April. Malo was finished on the 18th of April. I waited a week until all the citric was gone and sulphed it on the 25th of April. Then we started the analysis approximately 2 weeks after that. Okay, I don't think we have any further questions, so thanks for tuning in today. If you've got any further questions, please don't hesitate to contact us. As I say, we don't have time today to talk about control of scale and mealybug in the vineyard, and I'm not the expert there anyway. We've got a very experienced viticultural team here, so contact us through the normal way through the AWRI help desk. And thanks once again.